Welcome, dear students. Today we'll talk about cell visualization. In the previous session, we have understood about how we measure cells and the values in which we measure microorganisms as well as the components that are present within them, the biomolecules, and the various ways in which we understand how cells interact to form organisms. In today's session, we'll understand how we can visualize cells and microorganisms. The first method is through bright field microscope. This is used for unstained specimen organisms or cells which already contain pigments. So through that, we can easily identify any organism or any structure that is present within them. For example, the presence of chromoplast in case of certain plant cells, or the presence of certain granules which create a phosphorescent appearance or color in the cell. So through that, we can easily identify certain organisms. Or certain cells may already have high contrast, so they can visualize without any stain as well. In contrast, another method is phase contrast method, wherein we enhance the contrast by adjusting the light in such a manner that the cell is visualized with each and every part getting more contrast from the surrounding, improving the manner in which we distinguish the surrounding structures. In this case, we apply this method for unpigmented cell, although still we might not be able to see particular type of structures which may not be having sufficient amount of uh, ability to be seen in this particular method. Okay, so this basically happens in the manner in which the light is bending through these particular spaces, and that way the contrast is improved. Next is bright field or strain microscopy. This is the one that we often use in the labs as well, whether light microscope or compound microscope, where we use particular dyes or stain, which particularly in our scenario for most of the biology labs target nuclear, and that makes the nucleus visible under the microscope, and the surrounding cytoplasmic structures are also stained slightly less colored in comparison to the nuclear, but that improves the contrast between the nuclear and the surrounding structure. Okay, so next is the differential interference contrast. This is again based on your method of where your light interacts. So through that, we can again improve slightly the contrast and visualize cells in a better manner. Then the most common ones are the methods which are dependent on fluorescence. So what is fluorescence basically? You might have seen this thing happening when you take certain chemicals and apply blue light to it, and then we see that the chemical or the particular structure is glowing. For example, the light that is emitted by your fireflies, or the light that is seen on a scorpion when we use the blue light to glow it in the dark. So that is fluorescence. Fluorescence, in a very nutshell kind of manner, is when a particular wavelength of light falls on a particular structure, usually the pigment or any other material. A part of this light gets absorbed, and a lesser energy but a higher wavelength light is emitted out. We need to understand that by this equation, we know that our energy is inversely proportional to the wavelength of light. Okay, so a smaller wavelength of light, usually violet, blue, etc., are am- uh, absorbed by a particular pigment, in this case, which is often called as fluorophore. This fluorophore is then emitting certain wavelength of light of higher energy, of a higher wavelength, but of lower energy. And then through that, you're able to see certain color emitting out. Okay. So here there are two terms that we often use in the case of fluorescence. One is your absorbance, which wavelength is being absorbed, and then the next is emission. You might be wondering now, okay, I can get that through this. I can allow a pigment to be absorbing blue colored wavelength and then emitting out red. But then how can we obtain, say, blue color or a violet color? Because they themselves are of a lower wavelength. In that case, we apply the electromagnetic radiation from the non-visible spectrum or non-visible spectrum, that is your UV rays. So we emit the, we target the pigment with a UV wavelength that is absorbed by the pigment and then it emits out light in say blue or higher wavelength visible region. So that is how fluorescence basically works. And we use these special chemicals which are called as fluorophores for visualizing. Now, confocal works on that similar principle of using fluorophores. In this case, just the advantage is that the depth increases, the resolution increases, and so that you can distinguish neighboring structures quite easily. Okay. Now, there are these various cell visualization methods that we have covered. If we want to look at structures just with the naked eye, we can use a simple magnifying glass, but that will only help you visualize organisms which are in between the range of millimeter and micrometer, which are quite big enough to be seen by magnifying glass, such as a hydra. However, for an even smaller organism or a cell, we will require light microscopes or even better microscopes to visualize organism even better manner. Fluorescent microscopy still has certain limitations, and for that reason, we use electron microscopy whenever we have to look into organelles or whenever we have to look into bacteria or even smaller structures such as viruses. Now, think about what we just already discussed. What is fluorescence? And you can pause this video to explain in your own words what fluorescence is. Then think about the examples of fluorescence that exist in your daily life. How does fluorescence help in cell visualization? Now, also explore how is fluorescence different from another phenomena that is phosphorescence. To give you a clue, phosphorescence is what you observe in the star, moons, etc. that you often buy and stick them on your walls and they give a very green kind of sheen in your dark room. So think about what phosphorescence is and what fluorescence is. Another hint I can give you about phosphorescence is what you often observe in the yellow colored tape that is often placed on certain roadside materials or even on materials which are kept on the side of the road whenever you are driving the car at night and it glow up when headlight strikes on them. So think about how this process of phosphorescence is different from fluorescence. You can pause and think and search about it, or you can continue to the next part. Now, electron microscopy is one method in which we can understand cells even in more depth and in more detail because it gives even better information. In this case, you're not using light, but instead you're bombarding your specimen with particular type of metals or in a stream of electrons through which you're able to visualize the cells in much more clarity and higher resolution and more detail. For example, the images shown here is that of a cancer cell or a pollen grain, just still in the range of micrometer. But you can also see that here intestinal microvilli or your mitochondria are also shown, which are of the range of approximately somewhere between nanometer and micrometer. So even small structure like these, we can also see. Even better structures are also there. Even better instruments are also there, such as your X-ray diffraction or the other cryoscopic methods, which help you to visualize even macromolecules or the molecules that make up your cells. But that is not the scope of our discussion today. So this basically outlines how three ways of looking or understanding about our surroundings, the eye, the light microscope, and the electron microscope, helps you to visualize various objects. Your eyes can help you to look at clearly a particular person in front of you, or the width of your hand, or even certain part of your nail or your hair as well. But if you have to go into more depth of how these structures look like, you will have to use light microscopes. And even further, if you have to look into much more depth of how these structures are, you will have to go into the electron microscope. So the sizes are basically shown here, going all the way from 1 meters all the way to few angstroms. So when we write minus 10 as the power, it is used for angstrom. Minus 9 is for nanometer. Minus 6 is for your micrometer. Your minus 3 is for millimeter. And then 1 meter is also there. Okay. And then the same conversions on the above, you can also see are mentioned in the case of your nanometer and micrometer as well for you to understand better. In the next presentation, we will understand the differences between prokaryotic and eukaryotic cell. Research about various visualization methods and what are the advantages and the disadvantages. What are some of the new advancements that have happened in the cell visualization area and how to resolve old problems and what new endeavors they have opened in the research of biology. Till then, continue to read and learn. Thank you.